Good morning everyone, my name is Marsha and I'll be presenting my research project proposal this morning. My project is on the dosimetric evaluation of an intra-operative radiotherapy system. It will be both measurement based and a Monte Carlo modelling investigation. My supervisors include Pejman, Gabor, David and Martin. So I'll be talking briefly about the background of intra-operative radiotherapy as well as the Intrabeam device that I'll be making measurements on. I'll talk about the aims of my project, the significance and benefits, my project plan and some key publications. So IORT combines two conventional methods of cancer treatment, surgery and radiotherapy. It involves high radiation dose being delivered to a surgically exposed tumour bed. This is done in a single fraction and it normally takes about 10 to 30 minutes. So a key benefit of IORT is that it improves local regional control. And the benefits of this is twofold. You're more likely to kill the cancer cells and you're less likely to irradiate healthy tissues. It also reduces the need for post-surgical radiotherapy since you've killed the tumour cells in the tumour bed and there's less chance of regrowth. IORT is often used for breast cancer, pancreatic cancer and spinal metastases. I've got a quote here from the St John of God Subiaco Hospital and it reads, IORT is suitable for selected women with early breast cancer. It reduces treatment time from 3 to 4 weeks of daily radiation to a single targeted treatment administered while a patient is undergoing surgery to remove the cancer. So as you can see, these two benefits here are key for breast cancer, while the local regional control is the key benefit for pancreatic cancer and spinal metastases because of all the crucial organs surrounding the pancreas and the spine. So this here is the Intrabeam device that I'll be using. It's manufactured by Carl Zeiss, which is a German company. The maximum maximum voltage is 50 kilovolts, which means the effective X-ray energy is about 20 keV. So this machine differs to conventional low kilovoltage systems because of these 2 points here. The X-rays emitted are approximately isotropic, so uniform in all directions, and the X-rays are also very quickly attenuated in water and in tissue. We've also got spherical applicators, which are these things here that sit over the X-ray probe. And in the X-ray probe, you've got an electron beam that is accelerated down the probe and it hits a gold target here, where bremsstrahlung X-rays are produced. You've also got an internal radiation monitor, which measures the dose rate constancy. So if many X-rays scatter back up the drift tube and it reads 10% or more deviation, then it will actually cut off the radiation delivery. You've also got some shields. This is a spherical shield and that's a flat shield. And you can place them on the applicator, so not everywhere, not all the tissues in the patient are irradiated. So this is just a close-up of the X-ray production process. So you've got electrons coming down, they will hit the gold target here. And outside you've got a beryllium window, and beryllium is transparent to X-rays. This is a diagram of the actual device that we'll be using that we took photos of. Something new here is the photodiode array, which sits in here somewhere. So it helps measure dose isotropy and it will tune the electron beam if it needs to. And this is just the box that it comes in the top view of the X-ray probe. So the aims of this project include obtaining dose profiles for various spherical applicators, which are these things on the side here. We want to check for isotropy or uniformity in all directions. We want to compare our measured data with our Monte Carlo simulation and compare both of those with manufacturer reported data. So the significance and benefits of my project. Similar projects haven't been undertaken by the UWA medical physics team here before. It will be the first IORT project. However, Martin, one of my supervisors, has done similar evaluation of an Intrabeam device back in 2003. So it will be great to have his expertise and experience. The results of this experiment will help ensure safe and accurate IORT delivery to the patient. With better insight into dose isotropy, we can determine if shielding is needed in any particular direction to protect the patient. It should assist in the clinical process and save both time and labour. With a better understanding of the characteristics of the device, doctors can use it with more reassurance and more confidence, and this might result in more frequent use. If this were the case, it would reduce post-surgical radiotherapy needs for many patients and potentially shorten their treatment process. So this here is a map of my project plan. As you can see, the dosimetric evaluation of the Intrabeam will be twofold. We'll perform physical measurements as well as run a Monte Carlo simulation. So in terms of measurements, we'll be making measurements using an ionisation chamber as well as TLDs. The ionisation chambers are more accurate, but because the X-rays are so quickly attenuated, TLDs might be a better option because of their size and portability. And these results will be used to verify the film measurements, and film will give us a 2D dose profiles, which we'll then use to create a 3D dose map. On the other side here, with the Monte Carlo simulation, it will be run on eggs NRC, and these results will be compared against each other. And finally, they'll also be compared to manufacturer-reported data. The aim of this at the end is to make what we call an optimised treatment setup, which should hopefully help in the clinical process. This is a diagram of our experimental setup. As you can see, the film will need to be calibrated before it is used for dosimetry. The yellow sphere here is the applicator, which of course will come in a variety of sizes. These here are the TLDs, and as you can see, they'll be set out like this, so there's no interference or shadowing if they were aligned in a straight line. This is a summary of my literature review. The shaded boxes indicate the contents of each of the articles. As you can see, not all of them perform dosimetry with various applicators, but some of them do. Some of them use film and ionisation chambers, while some of them only use TLDs. A variety of Monte Carlo software has also been used and proven useful, including eggs NRC, Geont4, Geont4 with gate, and MCNP. This is Martin's paper down here. This is one of the key articles, Moradi et al. So they simulated the X-ray source as well as the breast phantom on the MCNP transport code. They used 1.5 to 5 centimetre applicators. They also performed physical measurements using ionisation chambers and radiochromic film. So they found the mean X-ray energy to be around 19.45. The ideal one was 20 keV, so that's pretty close. However, with applicators sitting on the X-ray probe, the X-ray energy was higher due to beam hardening. They also found that a larger diameter applicator resulted in a greater skin dose. So for this one here, which had a treatment depth of 0.5 centimetres and a diameter of 5 centimetres, the skin dose was 60% of the entire prescribed dose. This here is a figure of their Monte Carlo simulation in red and their film measurements in black. So this was the dose to water surface and as a function of distance from the applicator stem. So as you can see, they agreed fairly well, except potentially at the edges here. And they believe that was due to either uncertainty in positioning the film or due to the response at the film edge. And this here is just the red line minus the black line. So it just shows the difference between the film and the Monte Carlo simulation. This is another key article by Shamsabadi et al. So they used Geont4 instead, and they simulated the bare probe without the applicators. So they simulated the electron drift tube, the gold target, the beryllium tip, as well as various spherical applicators. So the percentage depth data was compared to manufacturer-reported values. The manufacturer used a soft ionisation chamber and what they call the Intrabeam water phantom to perform these measurements. So they found that Geont4 was indeed suitable and useful for Monte Carlo-based commissioning of the Intrabeam device. So this here is a summary of their results. This is just the bare probe, while these are the various applicators. The solid line is the measurement by the manufacturer, and the dots are from the Monte Carlo simulation. So as you can see, there is fairly good agreement for all the applicators and the bare probe. The uncertainty for their Monte Carlo simulation was about 1%. Thank you for listening. Are there any questions?
Great timing. Thanks very much. Marsha, any questions?
It's only one. It will be a really nice project. If you want to compare later our measurements, what you measure in ion chamber and border tank, like PPDs and these kind of things, maybe a continuation of this one could be that simulating the actual ion chambers in Monte Carlo. Because again, we are really close to the source in a PPD measurement. It's not a point source, it's not a point detector, and maybe all the response of the ion chamber will be completely different than what we think if you use that kind of ion chambers for normal X-ray devices. But if you can achieve the first part and get the proper model and get your measurements good, then it will be a huge task.
Yeah, I think that's definitely a possibility, simulating the ionization chambers.
Any other questions?
Actually, I was going to have a similar question to Gabor. Like all the detectors are film, even you could simulate this. Translate both of them to water, for example, so you can skip that step and just simulate the detector itself. But another question. So I guess the couple. So one is, what do you know about the device in terms of its structure and how it's manufactured? And do you know sort of the details of the dimensions and things of the materials?
I think some of the dimensions are on here.
Yep.
So the Berlin window, which covers the electron probe, is 0.5 millimeters, and the probe itself is 3.2 millimeters thick.
And so what would you need to adjust in that to match the measured data? What do you think? Do you think you'll need to tune that simulation somehow?
To match the measured data.
That you're going to be comparing against?
So our aim would be to simulate this. So whatever we measure will be the same as whatever we simulate.
Right.
So we'll hopefully be able to simulate the electron probe with all the widths and the gold target and the beryllium tip.
Yeah. There's probably some parameters there, like the width of the electron beam, how uniform its profile is, and what its energy spectrum might be. There might be things you might need to play with to make the simulation match the measurement. The other thing is, at these energies, the material that the X-rays are going through will have a big impact on your dose that you achieve. So if you simulate the source in water, for example, dose to water would be quite different than if you simulate it in breast tissue. So what materials will you be actually simulating?
I think we'll be simulating whatever we perform our measurements on, which I believe is a bolus, tissue equivalent bolus here. So we'll have stacks of bolus with film in between.
Do you know what the bolus is made out of?
No, I'm not sure. I'm not sure yet.
That's something you'll need to find out so you can do that simulation accurately.
That's right. I think David had a question.
Okay, David, any questions from Zoom?
Yes, I have a question, Marsha. I like the table you made about the literature, and especially the comparison between who used which Monte Carlo software. But do you know how many hours of computation each of the points you showed in one of the slides take?
No, I'm not sure.
What's the computational cost?
I'm not sure, but I can find out.
Yeah, it'd be worth looking at how much CPU time do you need to build these results?
Yes.
Considering just Marsha is in semester 1, and she has got 3 more semesters to go. And just start classes. Any other questions? All good? Happy? Okay.
lead.